Bismillahirrahmanirrahim. Assalamu alaikum. This is Dr. Huri Amir, and today we are going to learn about the digestion of various foods by hydrolysis. It is very important for all of us to know that how the ingredients which are present over there in our food, they are being digested by the help of GIT secretions, the enzymes, and the movements. Now let's talk about them one by one. First, we are going to talk about the hydrolysis of carbohydrates. Now, almost all the carbohydrates in our diet, that is polysaccharide or disaccharide or monosaccharide combination, and they are attached to one another by the process of condensation, which means that a hydrogen ion has been removed from one of the monosaccharides and hydroxyl ion has been removed from the next one, and the two monosaccharides then later on combine at these sites of removal, and hydrogen and hydroxyl ions they combine to form water. Now when carbohydrates they are digested, the above process is reversed and carbohydrates they are converted back into monosaccharides. Now how it is going to take place, that is that specific enzymes which are there in the GIT, they are going to return the hydrogen and hydroxyl ion back to the polysaccharide and going to separate the monosaccharide from one another, and this process is known as hydrolysis. So it is very important for all of us to know what is this term hydrolysis. So frequently it is going to be asked about that, define this process of hydrolysis. Hydrolysis. Now, hydrolysis of proteins. Proteins, they are formed from multiple amino acids which are bound together by peptide linkages. Now, at each linkage, there is a hydroxyl ion removed from one amino acid and a hydrogen ion has been removed from this other one. And then they are going to bound together by the process of condensation. And when digestion occurs, the reverse is going to take place, which is known as hydrolysis. That is, the proteolytic enzymes, they are going to return the hydrogen and hydroxyl ion back to protein molecules to split them into their constituent amino acids. Therefore, the chemistry of digestion is simple because in the case of all three major types of food, the same basic process of hydrolysis is involved. The only difference lies in the types of enzymes required to promote the hydrolysis reactions for each type of food. Now let's talk about the digestion of carbohydrates, the carbohydrate foods of the diet. Now there are only three carbohydrates, main sources, which are there for the normal diet. And what are they? They are the starches, the sucrose, the lactose. These are actually the large polysaccharides which are present almost all non-animal food like potatoes, different grains. So other carbohydrates ingested to a slight extent, they are amylose, glycogen, alcohol, lactic acid, pyruvic acid, pectins, dextrins, and minor quantities of carbohydrate derivatives in the meat. Digestion of carbohydrate in the mouth and stomach. When the food is chewed, it is mixed with that of saliva, which contains the digestive enzyme ptyalin, secreted mainly by the parotid glands. The enzyme is going to hydrolyze the starch into the disaccharide maltose and other small polymers of glucose, which contain 3 to 9 glucose molecules. The food remains in the mouth only a short time, so probably not more than 5% of all the starches will have become hydrolyzed by the time the food is swallowed. Digestion sometime continues in the body and the fundus of the stomach for as long as 1 hour before the food is going to mix with that of the stomach secretions. And later on, the activity of salivary amylase that is going to be blocked by the acid of gastric secretions. Why? Because the pH is going to fall below 4. On the average, before food and its accompanying saliva do becomes completely mixed with the gastric secretions, as much as 30 to 40% of the starches will have been hydrolyzed mainly to form maltose. Now, digestion of carbohydrates in the small intestine. Digestion by pancreatic amylase. Pancreatic secretion, like saliva, they contain large quantity of alpha amylase that is almost identical in its function with that of alpha amylase of saliva, but is more powerful several times. So, 15 to 30 minutes after the chyme is being emptied from the stomach and duodenum and is being mixed with that of pancreatic juice, virtually all the carbohydrates will have become digested. In general, the carbohydrates they are almost totally converted into maltose or other small glucose polymers before passing beyond the duodenum or upper jejunum. So see the starches in mouth by the alpha amylase and pancreatic amylase digestion, like you see. Then maltose, they are being 3 to 9 glucose polymers, and later on by maltase, alpha dextrinase, converted to glucose. This is how the um, draw and label the carbohydrate digestion table chart. Now, hydrolysis of disaccharides and small glucose polymers into monosaccharides by intestinal epithelial cells. The enterocytes lining the villi of the small intestine, they contain 4 enzymes: lactase, sucrase, maltase, and alpha dextrinase, which are capable of splitting the disaccharides lactose, sucrose, maltose, plus other small glucose polymers into their constituent monosaccharides. These enzymes are located in the enterocytes covering the intestinal microvilli brush border, so the disaccharides they are digested as they come in contact with these enterocytes. The lactose splits into a molecule of galactose and a molecule of glucose. Sucrose splits into a molecule of fructose and a molecule of glucose. Maltose and other small glucose polymers all split into multiple molecules of glucose. Thus, the final products of carbohydrate digestion, they are all monosaccharides. They are all water soluble and are absorbed immediately into the portal blood. In the ordinary diet, which contains far more starches than all other carbohydrates, combined glucose represents more than 80% of the final products of carbohydrate digestion, and galactose and fructose each represent seldom more than 10%. See how the digestion of proteins taking place by hydrolysis. The dietary proteins, they are chemically long chains of amino acids which are bound together by peptide linkages. A typical linkage is the characteristics are determined by the type of amino acids which are there and their arrangement in the molecule. Now, how the digestion of protein is taking place. First one, which is very important, it is important to know the protein digesting pepsin, the most important one in the stomach, and is active at the pH of 2 to 3 and is inactive like if pH is more than 5. And consequently, for this enzyme to cause digestion of protein, the stomach juices must be acidic. So this HCl secreted by the parietal cells are at a pH of about zero point 0.8, but the time it is mixed with that of the stomach contents and with secretions from the non-oxyntic glandular cells, the pH is going to become 2 to 3, which is highly favorable range of pepsin activity. One of the most important features of pepsin digestion is ability to digest the protein collagen, which is an albuminoid and is affected little by other digestive enzymes. Collagen is a major constituent of the intercellular connective tissue of meats. Therefore, for the digestive enzymes of the digestive tract to penetrate meats and digest the other meat proteins, it is necessary that this collagen is to be digested first. Consequently, in persons who lack pepsin in the stomach juice, the ingested meats they are less well penetrated and are poorly digested. Pepsin only initiates the process of protein, usually providing like 10 to 20% of the total protein digestion to be converted to proteoses, peptones, and a few polypeptides, and the splitting is actually the result of hydrolysis. Now, most protein digestion occurs in the upper gut, upper small intestine, and duodenum, jejunum, under the influence of proteolytic enzymes from the pancreatic secretion, and immediately on entering the small intestine from the stomach, the partial breakdown products they are being attacked by major proteolytic pancreatic enzymes trypsin, chymotrypsin, carboxy, polypeptidase, and proelastase. Both trypsin and chymotrypsin split the protein molecules into small polypeptides. Carboxy polypeptidase then cleaves individual amino acids from the carboxyl ends of the polypeptides. Proelastase, in turn, is converted into elastase, which then digests elastin fibers, which partially holds meats together. See? Digestion of peptides by peptidases in the enterocytes that line the small villi of gut. The last digestive stage of proteins in the intestinal lumen is achieved by the enterocytes lining the villi of small gut. These cells they have brush border having hundreds of microvilli projecting from their surface. In the membrane of each microvilli there is presence of multiple peptidases protruding through the membrane to the exterior, where they become in contact with intestinal fluids. There are two types of peptidases like amino polypeptidase and several dipeptidases, and they succeed in splitting the remaining larger polypeptides into tripeptides and dipeptides. And both amino acids plus the dipeptides and tripeptides are easily transported through the microvillar membrane to the interior of enterocyte. Finally, inside the cytosol, there are multiple other peptidases which are specific for the remaining types of linkages between the amino acids. Virtually, all the last peptidases and tripeptides are digested to the final stage to form the small to a single amino acid, and then they are going to pass to other side and ultimately into blood. More than 99% of final protein digestive products which are absorbed are individual amino acids, and with only rare absorption of peptides and very, very rare absorption of whole protein molecules. Even these few absorbed molecules of whole protein can sometimes cause serious allergic or immunological disturbances. C. The digestion of fats. The most abundant fats of the diet are the neutral fats, triglycerides. Each molecule is composed of a glycerol and 3 fatty acids. In the usual diet are some phospholipids, cholesterol. The phospholipids and cholesterol esters, they contain fatty acids and therefore can be considered fats. Cholesterol, however, is a sterol compound containing no fatty acid, but it does some exist to have the physical and chemical properties of fats, and it is metabolized like that of fats. Therefore, the cholesterol is considered from a dietary point of view as a fat. So why cholesterol is considered to be a fat? Digestion of fats in this intestine. A small amount of triglycerides is digested in the stomach by lingual lipase, which is secreted by the lingual glands in mouth and swallowed with saliva. Amount of digestion is like less than 10%. And later on in small gut, digestion continues. The first step is emulsification. By bile acids and lecithin. First step is in fat digestion is physically to break the fat globules into small sizes so that the water-soluble digestive enzymes can act on the globule surface. This process is called emulsification of the fat, and it begins by agitation in the stomach to mix the fat with the products of stomach digestion. Then most of the emulsification occurs in the duodenum under the influence of bile, the secretion from the liver that does not contain any digestive enzymes. However, bile does contain a large quantity of bile salts as well as phospholipid lecithin. Both of these, but especially the lecithin, they are extremely important for emulsification of fat. The polar parts of the bile salts and lecithin molecules, they are highly soluble in water, whereas most of the remaining portions of their molecules are highly soluble in fat. Therefore, the fat-soluble portions of these liver secretions dissolve in surface layer of fat globules with the polar portions projecting. The polar projections, in turn, are soluble in the surrounding watery fluids, which greatly decreases the interfacial tension of the fat and makes it soluble as well. When interfacial tension of a globule of non-miscible fluid is low, the non-miscible fluid on agitation can be broken up into tiny particles far more easily than it can when the interfacial tension is great. A major function of bile salts and lecithin, especially the lecithin in the bile, is to make the fat globules readily fragmentable by agitation with the water in the small bowel. This action is the same as that of many detergents that are widely used in household cleaners for removing grease. Now, each time the diameter of fat globule are significantly decreased as a result of agitation, and the total surface area increases manifold. Now, because the average diameter of fat particles after emulsification has occurred is less than 1 micrometer, this represents an increase of as much as 1,000-fold in total surface area of the fats by the emulsification process. The lipase enzymes are water-soluble compounds and can attack the fat globules only on their surfaces. Consequently, this detergent action of the bile salts and lecithin is very important for digestion of fats. Triglycerides are digested by pancreatic lipase. By far, the most important enzyme for digestion is pancreatic lipase, present in enormous quantities in pancreatic juice, enough to digest within 1 minute all triglycerides that it can reach. In addition, the enterocytes of the small intestine contain additional lipase known as enteric lipase, but this is usually not needed. C. The end products of fat digestion, they are free fatty acids. The bile salts they form micelles which accelerate the fat digestion. The hydrolysis of triglycerides is a highly reversible process, and therefore accumulation of monoglycerides and free fatty acids in the vicinity of digesting fats quickly blocks further digestion. But the bile salts, they play additional important role of removing the monoglycerides and free fatty acids. So this occurs. Bile salts, when in high concentration, they have propensity to form micelles, which are small cylindrical globules which are 3 to 6 nanometers and composed of 20 to 40 molecules of bile salts. So they develop. Why? Because the bile salt molecule is composed of a sterol nucleus, which is highly fat soluble, and a polar group, which is water soluble. The sterol nucleus is going to help for fat digestion, forming a small fat globule in the middle of a resulting missile with polar group of bile salts projecting outward. Now, because these polar groups are negatively charged, they allow the entire missile globule to dissolve in water of digestive fluid and to remain in stable form until the fat is absorbed into the blood. The bile salt micelles also act as a transport medium to carry the monoglycerides and free fatty acids, both of which would otherwise be relatively insoluble to the brush borders of the intestinal epithelial cells. Therefore, the monoglycerides and free fatty acids are absorbed into the blood as discussed later, and this process is known as the ferrying process. Now, digestion of cholesterol, esters, and phospholipids. Most cholesterol in the diet is in the form of cholesterol esters, which are a combination of free cholesterol and fatty acid. Phospholipids also contain the fatty acid within their molecule. Both the cholesterol esters and the phospholipids are hydrolyzed by the two other lipases, which are the cholesterol ester hydrolase to hydrolyze the cholesterol ester and phospholipase A2 to hydrolyze the phospholipid. The bile salt micelles, they play the same role in ferrying free cholesterol and phospholipid molecule digest states that they play in the ferrying monoglycerides and free fatty acids. Indeed, essentially no cholesterol is absorbed without this function of the micelles. So this is all for today's lecture. I hope all of you must read at home very nicely, precisely. So stay blessed and Allah Hafiz.